Good evening. Tonight we will be covering lesson 9.1, cellular growth. Two sections to this lesson, cell size limitations and the cell cycle. We'll start with cell size limitations. Why are cells limited to less than 100 micrometers in their diameter? There are two main reasons here. The bigger a cell becomes, the harder it is for its DNA to code all of that size, and the bigger it becomes, the more difficult it's gonna be for the cell to transport nutrients in and its waste out. And why is that? Because the bigger the cell becomes, the smaller the ratio of surface area to volume. So what does that mean? The volume of the cell grows faster than the surface area. And this, in turn, leads to a difficulty in transporting substances. Most of the substance transfer inside or in and out of the cell is done either by the plasma membrane, the cell membrane, because it's selectively permeable, or done by motor proteins which also pull it along the cytoskeleton of the cell. So no matter what, a bigger cell has to transport a longer distance. While in difference, a small cell can maximize this because it has to cover way less distance. While it is possible, it's much less efficient for a large cell. Another issue that arises is cellular communication. Cells use proteins, specifically called signaling proteins, to communicate different organelles, different parts of the cell. Now, cell size affects this protein's ability to move between here and there, therefore it limits cellular functions. The cell isn't able to control itself correctly. For example, here, let's say the ribosome wants to make a certain kind of protein. Now the DNA has this protein coded. We want to send the signal to the ribosome, but in a big enough cell, the signal never makes it. By the time the signal is there, the cell has already run out of this crucial protein and the cell has died. So, as we can go over the topics we covered, ratio of surface area to volume, transport of substances, and communication. Now, to the other half of this lesson. Cells get a choice at a certain point to either stop growing or divide. Divide being reproduction. Most cells choose to divide so that they may sustain more and more numbers, but certain cells, like muscle and nerve cells, they do not divide. They stop at a stage called the G1 stage. We will be talking about that later on. Cell division is how they reproduce, and this whole process is called the cell cycle. Now, the cell cycle has three stages. Stage 1, interphase. Stage 2, mitosis. Stage 3, cytokinesis. Interphase is broken down to 3 stages, mitosis down to 4, which will be in the next lesson, and cytokinesis, no stages. We will start with interphase. Interphase is the longest part of the cell cycle and is comprised of 3 stages, G1, synthesis, and G2. G1 and G2 stand for gap 1 and gap 2. In G1, the cell functions normally. It grows, it does all its needed functions to survive, and it is the longest stage it stays in. In synthesis, which is the second stage, it copies its DNA and prepares for cell division. In G2, third stage, it prepares for the division of the nucleus itself. So in synthesis, it's preparing for the division of the cell. In gap 2 or G2, it's preparing for the division of the nucleus. G1, as we mentioned, normal functions, preparing to replicate or copy the DNA. Synthesis, the cell copies its DNA, and we will delve deeper into that process. So it doesn't simply just copy the DNA. It takes the chromatins, which is how the DNA is in the nucleus, its relaxed form, and condenses it down to chromosomes, which is a condensed version of chromatin, and it is the genetic material needed for the next cell, the next generation. Chromatin, you could say, condenses, statins. It's like you packing your lunchbox, for example. In the third stage, it does its normal functions and it prepares for division of the nucleus itself. We will dive more into that in mitosis later on. In mitosis, it divides. You don't need to worry about this just yet. This is for next lesson. Now, cytokinesis. Cytokinesis is the division of the cytoplasm. You could tell from the name cytokinesis, cytoplasm. And it happens by a sort of a ring appearing at two ends in the middle of the cell, and it keeps tightening until the two cells are two separate cells. It happens after mitosis, as we mentioned previously. Now, the place where this ring forms, we are gonna call this the cleavage furrow. Prokaryotes, on the other hand, have no nucleus, and therefore the reproduction method has a different name. We call it binary fission. You don't need to think much about it, just know that it's how prokaryotes reproduce, while eukaryotes use the cell cycle. So even though it's prokaryotic cells, they don't use the cell cycle, they use binary fission. And that is all for today.